Dear student, today we are going to discuss application of biotechnology. First, in the field of agriculture, there is a green revolution. The green revolution related to the agriculture. The main aim of the green revolution to develop a good variety of the crops and increase the productivity. So, in the green revolution, that better agricultural practices and develop good tools and technique which improve the agriculture methods. Next one, GM crop. GM crops are called genetically modified crop. Those crops in which their gene are altered or changed due to the insertion, that is called genetically modified crops. So genetically modified crops having advantage over the conventional crop because they are insect and pest resistant. Insect and pest resistant crop, less use of chemical fertilizer, high nutrient value, biotic and abiotic resistant. Okay, like drought resistant and some insect and pest resistant. So that is the advantage of genetically modified crops. So there is a one genetically modified crop like bt cotton, bt brinjal, bt tomato, that is genetically modified crop. So bt crops, bt crop. So bt crop is a GM crop or genetically modified crop in which bt gene, bt stand for bacillus thuringiensis, thuringiensis thuringiensis. Bacillus thuringiensis is a bacteria in which this bt toxin gene are isolated. This gene isolated, bt toxin. So this gene are isolated and this bt toxin gene produce a protein that is called bt toxic protein. So this protein are produced, that is a toxic protein which cause the death of the insect. How this bt toxin protein isolated from the bacteria Bacillus thuringiensis and when inserted into the plant cell by recombinant DNA technology, and when the insect ingests the plant material, this toxic protein cause kill of the insect. So this Bt toxin first present in the form of protoxin. Protoxin means inactive toxin. So this Bt toxin not cause effect of the bacteria. Why? Because it mainly first present in the protoxin or inactive toxin. So it not cause any harm to the bacterial cell. When the insect ingest the material, this protoxin converted into the active toxin. How? Because the pH. Every enzyme, every protein required a pH and temperature to their activity. So the pH of gut is alkaline of insect. So due to the solubilization, this protoxin converted into the active toxin, and this active toxin cause death of the insect. Clear? Important point. So important point, a Bt crop is genetically modified crop. This is Bt gene isolated from the Bacillus thuringiensis. This is insect resistant crop. And insect like lepidopterans like fireflies, like army worm, mosquito, beetles, and house flies. That is insect resistant crop, and they having Bt toxin gene are not affected to the bacterial cell because it present in the inactive form called protoxin. When insect ingests the plant material, due to the gut alkaline pH, this is converted into the toxic inactive toxin, active toxin, and cause disease of the cell. So that is Bt crop. The gene for the Bt toxin called cry gene. Okay, cry gene. So cry gene encode a protein called toxic protein. Next one, that is RNAi. Very important. RNA, you know, ribonucleic acid. I means interference. RNA interference. What is this? RNA interference is a method of cellular defense. Cellular defense method in eukaryotes. किसमें होता है? यूकैरियोट्स में how this RNAi method silent the silence mRNA, particular mRNA in cell mRNA को साइलेंट कर देता है. हाउ फ्रॉम द जीन फ्रॉम द डीएनए दिस फॉर्म कॉम्प्लीमेंट्री डबल स्टैंडर्ड mRNA that cause silencing of specific mRNA and protein not formed. So that is RNAi method to give the resistance against the nematode infection. निमेटोड इन्फेक्शन से रोकने के लिए आरएनए मेथड करते हैं. लाइक मिलेडोजाइन इग्निशिया दैट इज निमेटोड from which RNA interference RNA method we can use by the formation of complementary double stranded DNA RNA, because generally RNA is single stranded and they express and forming the translation and form the protein. But when this RNA form complementary double stranded RNA from the DNA, that particular mRNA does not form go for the translation and protein cannot form. So nematode infection for the RNA method, that is cellular defense method to resist against the nematode infection in tobacco plants. Very important. Next one, that is gene therapy. This is agriculture we done. Next one, that is gene therapy. So what is gene therapy? Gene therapy, many of the hereditary disease, many of the autoimmune diseases like cancer, sickle cell anemia, PKU. So particular gene mutation are due to the defect of the gene, particular disease are occur. So that replaced by the functional gene, non-functional gene replaced by the functional gene. That method is called gene therapy. So first disease that is treated by the gene therapy, that is ADA, adenosine deaminase gene. This adenosine deaminase gene form adenosine deaminase enzyme. This enzyme important for the proper immune function. So when there is a mutation in the adenosine deaminase gene, mutation is take place, then this adenosine deaminase important for the immune system. So when the mutation is adenosine deaminase gene, then lymphocyte cannot form. Lymphocytes cannot form. Lymphocyte cannot form. So that is adenosine deaminase. So what we do for the treatment of this disease? There is many method like bone marrow transplant, enzyme replacement method. You give the injection of this enzyme. So and lymphocyte culture, in vitro culture of the lymphocyte from the blood cells of patient. lymphocyte बनाएंगे in vitro condition में. But these method are not permanent. Permanent method that bone marrow transplant during the embryonic time में bone marrow transplant करेंगे which carry the adenosine deaminase gene. Important. So gene therapy, adenosine deaminase important for the proper immune functioning and help in the formation of lymphocyte. So if the lymphocyte doesn't have, so immune system get weak. So adenosine can treated with bone marrow transplantation, enzyme therapy and lymphocyte culture outside the medium. So that is treatment for adenosine. Okay, next one, insulin production. So as you know that insulin, that is hormone is produced by beta cell of pancreas and maintain the blood glucose. So first you isolated this insulin gene from the cattle or any sources. So isolate the gene from source, isolated gene is isolated, insulin gene, and this gene are inserted into the vector by using suitable vector. This gene is isolated. So this gene having two band alpha and beta chain. So this alpha and beta chain are going to inserted. This is alpha, this is beta chain of the gene. Then this alpha and beta chain are isolated. Then after the insertion into the host bacteria cell, they separately form alpha chain and beta chain. Okay, this alpha and beta chain we are separated and obtaining alpha chain and beta chain and joined by the disulfide bond. So that is mature insulin. mature insulin. But there is a problem is that before the mature insulin, there is a pro-insulin. Pro-insulin. So what happened in the pro-insulin? In the alpha beta chain, along with there is a extra peptide called C peptide. C peptide. What is C peptide? So pro-insulin having C peptide. So before formation of mature insulin, this C peptide should be removed. Then this form mature insulin. First Eli and Lilly form the mature insulin. That recombinant method, that is called humulin. Recombinant insulin called humulin. Okay, beta. So that is insulin. So what we do, first we isolate the insulin gene, insulin having two chain, alpha and beta chain, and then introduce into the cloning vector. They multiply into the host bacteria, and then isolate, obtaining the gene of alpha and beta chain product protein. This is insulin is a protein. So then alpha and beta chain in pro-insulin having extra peptide called C peptide. This is removed, and then alpha and beta chain joined by disulfide bond. Okay. This is gene insulin. Next one, molecular diagnosis. So molecular diagnosis में हम generally use करते हैं बेटा PCR and ELISA. So PCR you know the polymerase chain reaction. अगर if you detect the infection, early infection and antigen, so you can do the PCR, polymerase chain reaction. If early detection of any antigen or pathogen, you can code earlier. So by PCR what you do, if the antigen, if their gene is there, they amplify their gene and you can easily identify the presence of particular antigen by the PCR. Okay, and by using PCR mutation you can also detect. And ELISA, that is for the HIV/AIDS. So ELISA mainly based on the antigen antibody interaction. So if there is any antigen in the your body, antigen mean foreign substance, then you do the ELISA, enzyme linked immunosorbent assay. So like this is a antibody, this is antigen. So that bind to the antigen and give the chromogenic substance color appear. So you can easily detect the presence of antigen in the body. So that is a method of detect the any antigen, that is molecular diagnosis. So revise all the points important for your exam. Thank you.